to this audience. Then with the glaucoma, I am going to focus on the primary and the angular glaucoma. So here, I think a concern with the genetic study, we will focus on what are the factors involved in glaucoma, as well as the main target is for each and every patient we are supposed to check the internal pressure, central column thickness, and the cup disc ratio. And in 1999 or 2000, I think they identified the candidate gene for glaucoma by University of Iowa group, Dr. Edwin Stone. That's what all day we mentioned many articles. So here, I think in that way, what are the cause or how the glaucoma is forming? There are three factors. The factors determining intraocular pressure is the main cause. So here, I think the overhead of the aqueous humor production by ciliary body, resistance to aqueous outflow across the TM, and then the level of epididymal vein pressure. So the second one we should focus on the research, because it is because of the outflow there is a complication. It is increased the intraocular pressure. So obviously the patients, they lose the peripheral vision, and then they will have only the central vision. So once the candidate gene is identified, especially the myosin, long back, 50 years back, that is called the myosin, that is present in the chromosome number 1. So people thought there is only one gene for glaucoma. I think if you screen 100 primary open-angle glaucoma patients, only they are positive for only 5 patients for the particular gene. It means, so rest of the 99 patients, they are negative for myosin. It means the additional candidate genes will be there for as the candidate gene for primary open-angle glaucoma. So far, if you see the Human Genome Project, we have 25,000 genes in our body, and then that is taking care of the entire function of the body. So among the 25,000 genes, 547 genes, I think that is approximate number, 547 genes that is present in the is responsible for ocular eye diseases. And part of them is retina. Part of them is retina in the sense 185 genes are already candidate genes for retinal diseases, and then around 200 loci is identified for retinal diseases. Rest of them is maybe some other diseases. Here, I think I listed out many genes. I think I don't know the pointer. So here, I think as Aldo mentioned, right now the technologies have been improved. The people did the entire genome-wide association studies, and then they identified not only the myosin gene, there are other 3 candidate genes for glaucoma. That is, it means, as I mentioned here, there is the myosin, optineurin, and then WDR36. These are the main 3 candidate genes. So rest of them. So if you see here, it means what I'm trying to see here. So 15 years back, people thought there's only one gene for glaucoma. Right now, it's not a single gene cell disease. It seems to be as a complex disease. It may be like a diabetic retinopathy or AMD or like that. It's a very, very complex. It means many genes are involved because so many other factors, as I mentioned. So we have to look into many different angles. What is the cause of the central corneal thickness, and then optic nerve damage, everything. There are other factors responsible for the glaucoma. That's what the gene is vast. It seems to be right now the glaucoma is a very complex disease. So here, so far, more than 30 loci identified for primary open-angle glaucoma, different ethnic groups. They are all listed here. And this is in addition to the additional candidate genes, some other genes are associated with this kind of factors. So that's what, these are the reports right now available, what are the causes for primary open-angle glaucoma. And this is the pathway I mentioned, and this is the very, very complex pathway primary open-angle glaucoma. If you see the left-hand side, 15 years back we understood about that particular pathway, WR36 and everything. So now it seems to be is a very, very complex. We have to deal with either through the proteomic studies and other things, then only we can able to understand what is exactly the pathogenesis of primary open-angle glaucoma. So I think these are the genes associated with the human. I think I don't go in detail. Maybe I'll come to briefly about the primary open-angle glaucoma. That is called the myosin gene. So this is is a very 3 exons, and then if you see the exon 3, the mutation, the hard spot of the mutation is there in the exon 3. And people have been screened all over the world. More than 180 variants is already identified in primary open-angle glaucoma. And these are the mutations. What we did in India, so in our Aravind Research Eye Hospital and in my lab, so I think we did a different kind of screening, as well as our aim is to make a DNA chip for diagnosis. In the sense, so L.V. Prasad Institute and Sundaresan and Aravind, so we combined targeted to why not we make a DNA chip for diagnosis. So that's what we applied to the government of India, and we collected more than 1,000 samples. And then, so because to come to when you make a chip, I think it should not represent only the southern part of India, and then we collected some of the samples from AIDS too. So we collected all the samples, and then we did the myosin screening, and then this is the report, So this is, from all the four centers, and then, the Mayak mutation, all the 1,000 samples, and what is exactly mutations is present in the primary open angle glaucoma, as well as, there is another gene that is called the CYP1 gene, that is the candidate gene for congenital glaucoma. I don't want to talk about the congenital glaucoma, but it is having a modifier effect, So suppose that is called the modifier gene, it is associated with the primary open glaucoma. So when we screen the primary open glaucoma to this particular gene, we can able to identify the mutation too. That's what we call it, these two genes and how it is involved in the pathogenesis. So like that, our aim, we come up with some part of mutations that is common in Indian population. So that is published in long back. And if it come. This is, these are the information we already published from Aravind, this particular data from Aravind. So we analyzed more than 200 primary open glaucoma patients. You can see those two, whatever I highlighted, those two mutations as very, it made a significant interaction with the primary open glaucoma. If you see that the 367 glycine arginine mutation, so when you see the patient, the case is very thick because right now he is about 55 years old. The onset of the disease is 40. So I think when we did the entire family, so he is the patient is the elder brother, and then the younger brother is even 15 years lesser than him. So when we screen, and then the patient is as a 55 years old, he is positive for this mutation, and then obviously we can call it with the pathogenesis. But the classification of the POAG. You can divide it the juvenile and the primary open angle glaucoma. More than 40 years is a POAG, but the younger brother in the family is, we identified the mutation, but he didn't get the disease. It means there is no glaucoma for him. Definitely he is prone for the disease. So that's what we did a counseling. So he working as a driver in the local transportation, and then we put it in a different way. I think we should not make the patient very panic. So I think he may prone for the disease. Why not to come to the hospital for the regular checkup? So like that, we analyzed the entire family for this particular thing. And another one, that the T377 methionine, that particular mutation is a very interesting mutation. That is, through mutation you can able to identify the founder's effect. Founder's effect in the sense how, suppose one particular group or people migrated from one continent to another, like British people migrated to Australia. Suppose if you deal with the one particular disease, glaucoma in UK and Australia, definitely you can identify the founder's effect in concern with the mutation. The same way, this 377 mutation, this is a kind of, we identified the founder's effect once we publish the data. I think all over the world in US, I think why not we confirm this is the founder's effect for this particular disease. We have another publication along with other US group. That is one particular contribution we made for the primary open glaucoma. And then again, the secondary complication, because if you see, I think I'll touch about a little bit the secondary complication, pseudo exfoliation. So some people, they have only pseudo exfoliation alone or some pseudo exfoliation with glaucoma. So we are recently our paper is accepted in JAMA Ophthalmology. I think it is going to publish next month. So I think we did screening, it is the Loxel one. So whatever I highlighted, that is the primary mutations that is correlated, is giving a positive correlation to the Indian population, as well as we screen the entire the promoter regions of this particular gene. And these are the testing, and it is possible that it's a congenital glaucoma or it is a normal tension glaucoma, that is possible to screen these particular genes. It is possible. And then very recently, I think as I mentioned, the genome, there is no candidate gene for primary ankylosing glaucoma. So till last year, the candidate gene is not yet identified. But in the Singapore Research Institute, there's a Angten group. They screened more than 8,000, 9,000 samples. All over India also contributed. Sundaresan also contributed some samples, some 2,000 samples. So this is, I'm talking about the ankylosing glaucoma. They did the genome-wide association. It's a very expensive study, and then they did all the samples, like 10,000 samples all over the world, Hong Kong and China and everything samples, and then they identified the very first time, and the three genes associated with the primary ankylosing glaucoma. So that is that is a very recent report in ankylosing glaucoma, and we also replicated the study in Aravind, and then we published it very recently in IOBS, and that is the kind of replication study, and that is supporting whatever the previous result showing. That is, we prove yes, that is the particular gene is associated with the primary ankylosing glaucoma. And then it is a very recent report, and Gerald Watt, he asked me why not you talk about something related to gene therapy in glaucoma. So here I think obviously you should not concern with the DNA alone. You want to check the gene expression. And then right now, so I think this is from the Duke University. What they did, they took the cadaveri, take the glaucomatous and non-glaucomatous, the cadaveri after enucleation. You take the TM, trabecular meshwork, from the glaucomatous and non-glaucomatous, and then take the RNA, because that is represent the protein, and then using the high technology, and then they analyze all the proteins and everything. So here, I think this is a very recent report. That this is why I am telling. This kind of thing is obviously you want to identify the biomarkers or the gene that is responsible for the disease. This kind of experiments definitely it will give you a way, and we can able to identify the gene or the markers for the primary ankylosing glaucoma. This is an attempt. So if you see the conclusion, this is and then they are talking about the shedding. Suppose if you see the aqueous humor, it is shedding in the exosomes. That is also representing some of the proteins. So they are analyzing all those things in the proteomic analysis, and then they can come to a conclusion. And then this is already mentioned by Aldev. And suppose concerned with the any genes, I think we can some approved laboratories are there. You can send the sample for the genetic analysis. So he talked about coronary diseases. Again, I think I already associated with Dr. Edwin for the first 15 years, and they are doing a lot of research on glaucoma there, so you can go for diagnosis. And even Aravind, we studied more than 30 eye diseases. We are also performing some molecular diagnosis for many eye diseases too. And another one is concerned with the gene therapy. So before go for the gene therapy, and this is again from Duke University. So you're supposed to test the gene in the animal model. So here I think the study to the in vivo testing the lentiviruses delivering the gene to the trabecular meshwork. In the sense, so they injected the virus and the DNA, and then they put into the animal. So here see the expression. Suppose when the delivery, so they are, obviously we supposed to target with the TM, trabecular meshwork. So the expression of the lentivirus and the glaucoma thing. So it is expressing only the. I think I don't know the pointer. I'm sorry. So here. Okay, so this is the endothelium, and this is the cornea, and this is the meshwork. The expression is very high only in those places, and then when concerned with the cornea, the expression is not in the epithelium or in the stroma. It is only in the endothelium and then the meshwork. They proved using this one. I think the specific expression is there specifically in the animal model. They proved, and then this is a kind of gene therapy for glaucoma. It's a kind of review. In ophthalmology, I think they attempted for retinoblastoma for gene therapy, but it's not that much successful. Successful, but very successful gene therapy is for Lieberkranz amaurosis. That's what Sarada is going to talk about. So that is very successful story for Lieberkranz amaurosis. Concerned with glaucoma, I think many people are trying. If you see here, I think the recent laboratory studies utilize the gene therapy techniques to the lower the intraocular pressure and everything. See here, I think these tissue. Here only what kind of vectors you can use to deliver the viruses. Because if you take one particular virus, adeno-associated virus, there are so many types are there, which is efficiently to deliver the gene. That kind of experiment is going on, is very, very, very preliminary. People are trying on that. There is no success story for glaucoma. So when compared with liver amaurosis. And then, as I mentioned, in addition to the DNA, when you target with the DNA, and in addition to the DNA, you can use the biological fluid from the whatever you are dealing with eye diseases. Suppose if it is a cornea, corneal diseases, you can take tear and then you can analyze the proteomic analysis, compare the tear between the normal tear and the abnormal tear, the diseased tear, and then you can match with the pathogenesis. Like that. So we are also dealing with the primary diabetic retinopathy, glaucoma, and then other things. Suppose if it is a glaucoma, obviously you are taking the aqueous humor, or sometimes you are supposed to take the mesh one. Or if it is a vitreous, based on that, what are the biological fluid available, you can get the proteins, and then this is one of the modern approach to detect the biomarkers. Because concerned with the biomarkers, maybe I will take another 2 minutes. So biomarkers, suppose there is biomarkers in the sense, see the very successful biomarkers for nephropathy. I think you can detect the biomarkers in the urine. Concerned with nephropathy, there is a marker called microalbumins. So that microalbumin, even before the onset of the disease, the nephropathy, because the nephropathy the start around the 65 or 70 years. So the onset of that particular microalbumins, it will come through the urine at the age of 55 or 60. So it means that if you find out that kind of markers, definitely, because many of the eye diseases are like primary open, there is no symptom at all. So that's what people are trying for the biomarkers to prevent the eye diseases. This is a very recent article from American Journal of Scientific American. So right now they are talking about the personalized medicine. If you say just $5,000 or $1,000, your genome is ready. That's what Journal is talking about. So I think that is the future thing. But if you see in the bottom, so I think for researcher, you can go for the genome sequencing or the personalized sequencing. But concern with the patient point of view, it is very difficult. That's what the article says. I think for the clinician, obviously, without your support, we can't able to do research, and maybe that is in the handout, and you can go through that. Thank you very much.
Thank you, Dr. Sundaresan.